So the cochlea of the inner ear um, kind of spirals around um, with the central modulus, and but if you were to look at each of the, or if you were to look inside the tube, um, it actually has three compartments, namely the scala vestibuli, the scala tympani in the bottom, and the scala media. So it's much like the inner tubing of, let's say, a car tire. The scala media runs along the length of the cochlea, and the scala media compartment is what we call as a membranous labyrinth, kind of floating in within the bony labyrinth around it. So the scala vestibuli and the scala tympani are compartments of the bony labyrinth, while the scala media is a part of the membranous labyrinth. So there's two important membranes that separate the scala media from the scala vestibuli in the top and the scala tympani in the bottom, namely the resinous membrane that separates the scala vestibuli from the scala media, and then the basilar membrane that separates the scala tympani from the scala media. This basilar membrane is actually an important anatomical structure, especially because the organ of Corti lies on the top of the basilar membrane. And it's in this organ of Corti where those sensory end organs are structures that we call as the inner hair cells and the outer hair cells lie. It's a little bit more about this basilar membrane. It's about 35 millimeter long or 3.5 centimeter long, but since it spirals around the modulus, it's much smaller, looks much smaller. It's a very thin membrane, and in terms of breadth, it actually is narrower at the base, and it gets wider as you go towards the apex. So in this figure illustration, you can see that this is the base, and this is the apex, or the top of the cochlea. I know this is the base because this is where the oval window and the round window is. So the stapes actually sits on the oval window right there, and that's where the stimulus comes into the inner ear. So when the ossicular chain is moved by the sound hitting on the tympanic membrane, the stapes footplates move in and out of the oval window, moving the fluids inside the inner ear compartments. And the pressure that's created by this actually goes along the length of the cochlea till the very end, what we call as a helicotrema, that opening that connects the scala vestibuli to the scala tympani, and that pressure is dissipated by the round window over here. So while it's traveling, I should say, along the length of the inner ear, it stimulates different parts of the cochlear partition on the basilar membrane. And as I said, the basilar membrane is arranged such that it's narrower at the base and it gets wider as you go to the apex. And this is in contrast to the actual size of the cochlear duct. If we were to look at the bony labyrinth, you'll actually see that the cochlear duct appears wider at the base, and then it gets narrower as it goes to the apex. That's an interesting contrast, but it has a physiological significance we're going to be talking about in just a bit. It's this varying length of the basilar membrane that helps in separating the frequencies of this incoming sound. And when we talked about in acoustics how length and stiffness affects, if you recollect, we talked about how as length increases, the frequency, the resonant frequency, actually decreases. And then in terms of stiffness, as the stiffness increases, actually the resonant frequency increases, or the best frequency that this body can vibrate in increases. Okay. A little bit more about the anatomy of the basilar membrane. This basilar membrane is actually held by bony projections in this cochlear partition. We've got the spiral lamina towards the modular side holding or kind of pinching this basilar membrane, while we've got the spiral ligament at its outer or periphery holding the basilar membrane. And it's the spiral ligament and spiral lamina that kind of changes in length to make the basilar membrane narrower at the base and wider as it goes to the apex. Okay. And as I said, situated on the top of the basilar membrane or superior to this basilar membrane is this important organ of Corti. Here is another illustration of how the stapes is attached to the oval window, which opens into the scala vestibuli at the top. Scala vestibuli in the top, and at the round window opens into the scala tympani in the bottom, while the scala media lies in between the scala vestibuli and the tympani, and it's separated by this resinous membrane and the basilar membrane. Good. Again, in this illustration also you can see how the basilar membrane, that middle striated membrane over here, varies in length or breadth. It's more narrower towards the base, and it gets wider as you go towards the apex. So here, again, at the base, it's narrower and stiff. So we know that makes it responsive to the higher frequencies. So here, as a length is inversely proportional to frequency. So narrower membranes or narrower objects best respond to higher frequencies, while stiffness is directly proportional to frequency. So as an object gets stiffer, it best responds to higher frequencies. So at the base, since it's narrower and stiffer, the higher frequencies are best represented at the base, over here. At the apex, where it's wide and more slack or less stiff, it best responds to those lower frequencies. And those frequencies that we're talking about is that whole range of frequencies that the human auditory system can listen to. That's, in other words, 20 hertz to 20,000 hertz. So you can, technically, the highest frequency that we can respond, the 20,000 hertz, is represented at the very tip of the basilar membrane, the basal tip of the basilar membrane, while the lowest frequency that we can hear, 20 hertz, is on the apical tip of the basilar membrane. And all the other frequencies fall in between. Progressively, as you go from apex to the base, we move from low frequencies to higher frequencies. Again, in some of the discussions that I saw on the decibel, we talked about the wide range of hearing, and some had noted it's at 20 hertz to 20,000 hertz. Just to remind you that 20 to 20,000 hertz refers to the frequency range that we can hear. But even within that frequency range, each of those frequencies, we can hear a wide range of intensities or amplitude, and that's where the 10 to the power of 14 times come into play, and the need to compress that using the logarithmic decibel scale. Just a point to remember. So the frequency is actually the 20 hertz to 20,000 hertz, and we also talked about how we actually hear even better for those mid frequencies. So in other words, we're more sensitive to those mid frequencies than we are for very high and very low frequencies. Here's an interesting animation that talks about this frequency organization along the basilar membrane, a concept that we're going to be talking about a little bit later, known as tonotopicity. So tonotopicity refers to that frequency organization within any given structure in the auditory system, and it's kind of one of the important themes of the auditory system. It seems like nearly all the structures within the auditory system, uh, there is this tendency for the frequencies to be distributed spatially, separated along any given structure. And that's what we call as tonotopicity. Um, so the inner ear is such a fascinating anatomical structure. I talked about it, how it's as small as the nail of your pinky finger. Uh, so, and it But what's more important is the structure is even much smaller in dimension. So here the cochlea might be in dimensions, uh, but then the structures lie within the turns of the cochlea, are of the size of micrometer, but the actual magic happens in structures even smaller than that, namely those hair cells, and at the top of the hair cells, structures that we call as stereocilia, which are of the dimensions of nanometers. Nanometers. Nanometers are 10 to the power of minus 9 meters, while micrometers are 10 to the power of minus 6 meters. So micro is 1 millionth, is nano is almost a 1 billionth of a meter. So we're talking about real small structures. And that also explains why we had to wait till technology developed so we can actually look into those structures and understand these structures. So here is a scanning image of the cochlea, um, and this is probably a mammal cochlea because it's got a lot more turns than what we expect to see in humans. I think this is a guinea pig's cochlea. But nevertheless, you see the structures are quite similar to what we see in a human cochlea. So we're seeing those three compartments, the scala vestibuli, tympani, and the scala media. We're also seeing that central core um, known as a modulus. The modulus is a perforated structure because the auditory nerves fibers kind of enter through the modulus and they serve the different turns of the cochlea. They kind of extend into those hair cells and connect to those hair cells. And again, we're seeing the resinous membrane separating the scala vestibuli from the scala media. And then at the bottom, we've got the basilar membrane separating the scala tympani from the scala media. Another illustration of this cochlear partition, and the organ of Corti sitting on the top of the basilar membrane. Oh, I'm sorry, pardon my puppy barking his head off over there. So in this illustration, again, you're seeing the breaking down of the inner ear cochlea. So if we were to focus on each turn of the cochlea, you're seeing those three compartments with this organ of Corti lying at the top of the basilar membrane. And if you were to look more closely into this organ of Corti, it's when we're seeing those hair cells, um, namely the inner hair cells over here that lie close to the modulus or the central core over here. So we've got the inner hair cells right there. And then we've got three or more rows of the outer hair cells lying towards the outer edge of this cochlear partition right over there. So this is an image from what we call as an electron microscope. We're actually seeing how the hair cells actually look like. So you're seeing those hair cells over here. These are the inner hair cells. There's a row of the inner hair cells. In humans, there's only one row of the inner hair cells along the length of the cochlea, while we have three or more rows of the outer hair cells that are towards the outer edge of the cochlear duct. Um, in this illustration, you're also seeing this membrane that seems to have curled up, but in reality, this membrane kind of lies uh, kind of on the top of the hair cells. And this membrane is what we call as a tectorial membrane. Tectorial membrane. And it also plays an important role in the physiology of the inner ear. The tectorial membrane, in the undersurface of the tectorial membrane, the outer hair cells, stereocilia, are connected, while the inner hair cells kind of lie just below that, the tectorial membrane. The inner hair cells and the outer hair cells are separated by this tunnel known as this tunnel of Corti. Um, and actually the hearing nerves, the auditory nerves, traverses within this tunnel of Corti, and then they serve the different hair cells as they go to the apex. Another illustration of the inner ear and the hair cells and the organ of Corti. The reason why I provide so many illustrations is to kind of help you guys make an idea of how the inner ear looks like. And it always helps to look at different views and kind of breaking it down to understand the organization of the inner ear. So here you're actually seeing a blown up of the inner hair cell over there. And the top of the inner hair cells lie these even smaller hair cells, and these are the ones that I was talking to you about in the dimensions of nanometers, or 1 billionth of a meter. So these are rows of even smaller hair cells sitting on the top of the inner and outer hair cells. They're known as the stereocilia. They're the ones that actually pick up the fluid movement, and those hair cells convert that into a language that the nerves understand, namely converts it into electrical energy that's conducted by the nerves, and which goes higher in ultimately towards the brain. The stereocilia, stereo referring to sound, and then cilia referring to hair structures. So okay, so now we talked about how the fluid of the bony labyrinth is the perilymph, and the fluid within the membranous labyrinth is the endolymph. So the scala tympani and the scala vestibuli um, have this perilymph fluid, perilymph, peri outside, while the scala media, which lies between those two compartments, has this endolymph. Uh, these endolymph and the perilymph are separated by these membranes, the racemous and the basilar membrane, and they have different chemical compositions. And because of the different chemical composition, they also have different voltages to them. They have a different direct currents. The endolymph is high in potassium and it has less sodium in it, giving it a small positive voltage, namely that plus 80 millivolts. And relative to that, the scala vestibuli and the scala tympani have very low voltage. The perilymph has low potassium content, and then hence it has a low positive voltage. So the inner lymph is similar to the fluid that kind of bathes the central nervous system, namely the cerebrospinal fluid, while the perilymph is similar to the fluid that lies in the rest of the body, or what we call as extracellular fluids. So as I mentioned earlier, the inner lymph has a strong positive potential of plus 80 millivolts, and it's what we call as an endolymphatic potential. While the scala tympani and the scala vestibuli, which has a perilymph, has a much lesser voltage to it. So while the scala vestibuli has a plus 3 millivolts, a small positive voltage, the perilymph of the scala tympani has almost 0 millivolts. And all the structures surrounding these compartments actually have a negative potential. Um, okay. As I mentioned earlier, the organ of Corti has this inner hair cells, the outer hair cells, and some supporting cells that hold these hair cells in place. One of the important, a few of the important supporting hair cells are those inner and outer rods that actually form that tunnel of Corti that separates the inner hair cells and the outer hair cells. So here in this cross section of the organ of Corti, you can actually see the inner hair cells over here, and then the outer hair cells. You can also see the tectorial membrane that lies, kind of hovers on the top of this organ of Corti. And it's only recently they found out that the stereocilia that sits on the top of the outer hair cells are actually embedded on the undersurface of this tectorial membrane, while the stereocilia of the inner hair cells are not. And that apparently has a protective function. So when the basilar membrane moves because of the fluid movement, which in turn is because of sound impeding on the, well, eventually reaching the stapes and moving the fluids through the oval window, it's the stereocilia of the outer hair cells that are being deflected directly, but the inner hair cells are being deflected by the fluid that the outer hair cell stereocilia pushes. Okay. Why a protective function? Because now, since the inner hair cells are not directly connected to the tectorial membrane, if there's a really loud sound, it actually doesn't result in directly moving the stereocilia of the inner hair cells, but it's by indirect means of the fluid movement. And that's the reason why when we see individuals with what we call as noise-induced hearing loss, when they are exposed to loud sounds, you'll see that the outer hair cells are the ones that are affected first. And when we talk about the disorders of the ear, I'll actually show you some images where you can see after acoustical trauma or noise-induced hearing loss, you'll see that stereocilia of the outer hair cells are actually missing, are distorted, while the inner hair cells are only destroyed if this noise exposure was for a prolonged period. So there's all those inbuilt protective functions within the architecture of the inner ear. That's fascinating. So here's another illustration of the inner hair cells, IHC, and the outer hair cells, OHC. Here again, you can see that there's only one row of the inner hair cells, while there's 3 or more rows of the outer hair cells. Again, you're seeing this basilar membrane BM over here, and we can see this tunnel of Corti that separates outer hair cells from the inner hair cells. So situated on the top of the basilar membrane, as I said, are 3 to 5 rows of outer hair cells. So total, in average, we have about 12,000 to 15,000 outer hair cells. While we have only 1 row of inner hair cells, about 3,000 inner hair cells. So we've got this tunnel of Corti separating the inner and outer hair cells, with the inner hair cells being toward the medial end of the tunnel of Corti. In other words, it's towards that central core or modulus. Uh, just imagine another way of thinking about this cochlea is like a spiraling parking garage. Um, so towards the center of the spiral is those inner hair cells, and towards the periphery or the outer edge of the spiral is your outer hair cells. Well, it's the reason why it's called inner hair cells and outer hair cells. So these hair cells are really small structures. They're only about 0.01 millimeter long and 1,000th of a millimeter in diameter. But even smaller structures sit on the top of these hair cells. We call it the stereocilia. Okay, these inner hair cells and these outer hair cells differ um, in terms of their shape, the manner of innervation, how the auditor innervates these hair cells, and the organization of the stereocilia that sit on the top of those hair cells. So in this scanning electron microscope, you can see again the organ of Corti, the inner hair cells, the outer hair cells over here. Um, so you've got the inner hair cells over here towards the medial end, and then you've got the outer hair cells over here towards the outer periphery of the outer edge of the cochlear duct. And you can notice some difference in the shape of the outer hair cells and the inner hair cells. The outer hair cells are more like test tube shaped, while the inner hair cells are more like vase shaped. Um, so the inner hair cells are round shaped, while the outer hair cells are longer and thinner and more like test tube shaped. The inner hair cells stand alone, while the outer hair cells, if you were to look at that figure again, you'd see that it's embedded within one of the supporting cells that we call the cells of dieters. Also, there's difference between the organization of the stereocilia that sit on the top of the hair cells for the inner and the outer hair cells. The inner hair cells stereocilia are in a single row, while those of the outer hair cells are actually V or W shaped in some cases. Um, and apparently that has a physiological significance also. When we talk about the physiology of the inner ear, we'll talk about how the outer hair cells are responsible for stimulating the inner hair cells, because the outer hair cell stereocilia are those that are actually in contact with the tectorial membrane that sits on the top, and the inner hair cells are stimulated by the fluid movement that's caused by these outer hair cells. Now, with outer hair cell stereocilia shaped like a V or W, it's almost like cup shaped, and so it's more efficient in pushing the fluid towards the inner hair cells, much like what we do when we are trying to scoop water from the surface of a table. We kind of cup our hands, our palms, to effectively push the fluid. So apparently that's what's happening when those hair cells are being stimulated. The outer hair cells move um, and effectively push the fluid towards the inner hair cells. So here's an actual scanning electron microscope view of the top of the hair cells. So here we are seeing the inner hair cells, kind of almost the stereocilia almost like in a straight line, while those with the outer hair cells are actually more V or W shaped in some cases. And again, the inner hair cells are more medial, while the outer hair cells are more lateral on the top of the basilar membrane. Another illustration of the same point. So here the stereocilia of the inner hair cells in a straight row, while those of the outer hair cells are V shaped over here. And again, here also you're seeing the overall shape of the outer hair cells are more like test tube shaped. They're longer and thinner, and actually the inner hair cells are more like vase shaped. Actually, at the top of the hair cells, below the stereocilia, there's this laminar membrane known as a reticular lamina. It's a delicate membrane, and it's on the top of the hair cells, but below the stereocilia. So the stereocilia are actually embedded uh, on the top of the reticular lamina like that. Oh. Then we talked about the tectorial membrane. The tectorial membrane actually covers, kind of hovers above the stereocilia. Uh, and it's only recently that they found that the inner hair cells, stereocilia is actually not in contact with the tectorial membrane, while the stereocilia of the outer hair cells are actually embedded on the undersurface of the tectorial membrane right there. So the outer hair cells, stereocilia, are actually embedded, while the inner hair cells, stelia, are just below the tectorial membrane. There's a gap over there. So here you're seeing the tectorial membrane coming from the medial edge and kind of hovering at the top of this organ of Corti. And it's much more labeled in the schematic illustration over here. Okay, let's talk a little bit about the innervation of the cochlea. The cochlea is innervated by the 8th cranial nerve, the auditory vestibular nerve. And the auditory vestibular nerve is for majority it's an afferent nerve, or it's a sensory nerve. In other words, it's a nerve that carries information towards the brain, and that it's its main purpose, because the inner ear, for most purposes, is a sensory organ, so taking information towards the brain. But what was surprising is when anatomists discovered there's actually a small set of efferent nerves, or motor nerves, that actually bring in information to the inner ear. Um, so while we have majority of afferent nerves, or A sensory nerves, about 30,000, there's a small subset of efferent nerves, about 1,800. For a long time, anatomists didn't understand what was the role of those efferent or motor nerves in a special sensory organ like the ear. It's only recently they've understood that it plays an important role in the physiology of the inner ear that we're going to be talking about later. There is a difference between this innervation of the inner hair cells and outer hair cells. The inner hair cells um, receive a majority of the afferent innervation. About 95% of the afferent innervation comes from the inner hair cells. So obviously you can see that the inner hair cells play a very important role in cochlear transduction, because they're the ones that ultimately take the information to the brain. But outer hair cells seem to receive the majority of the efferent innervation, or the motor innervation. And again, it's only recently that we understood what role those outer hair cells play. So it's always baffled anatomists why we have a lot more outer hair cells than we have inner hair cells, while the inner hair cells are the ones receiving majority of the afferent innervation, or the sensory innervation. So the role of the outer hair cells has always been debatable, but now we've got a better understanding of what it does, and something that we're going to be talking about in the physiology in the next lecture. Also, not only the inner hair cells receive the majority of the afferent innervation, it also receives a condensed or intense innervation. A number of the afferent nerves innervate each of those inner hair cells. So it receives what we call as a many-to-one innervation. Almost about 20 afferent nerves innervate each of the inner hair cells. So it receives dense innervation, while only one afferent nerve innervates about 10 or more outer hair cells. So relative to afferent innervation, the outer hair cells receive much lesser innervation. So the inner hair cells are innervated about 20 afferent nerves. So if you were to describe that, we call that as a cell to neuron ratio. So each cell of the inner hair cells receives about 20 afferent nerves, while a single afferent nerve innervates about 10 outer hair cells. So if you were to calculate the cell to neuron ratio for outer hair cells, we have 10 to 1. In other words, 10 afferent nerves innervate each outer hair cell. But again, the outer hair cells receive the majority of the motor or efferent innervation. Again, that plays an important role in the physiology or the fine tuning um, of the traveling wave.